Good afternoon, everyone, and thank you for joining us today for our virtual public seminar, celebrating strong women in science. We have a very exciting lineup of talented speakers for you today, so thank you for joining us. It's wonderful to see so many familiar faces in the Zoom room, as well as some newcomers. Welcome to you all. And for those of you who don't know me, my name is Mara-Jean Tilley. I have the great privilege of heading up the Garvan Research Foundation, which is the marketing and fundraising arm of the Garvan Institute of Medical Research in Darlinghurst. I'll be your MC this morning. Before we begin, it's my humble privilege to pay respects to the Gadigal people of the Eora Nation, the traditional owners of the lands and waters on which the Garvan Institute is located. I recognise their continuing connection to land, waters, culture, and we pay our respects to their elders past, present and emerging. Today, Garvan and the Science Teachers Association of New South Wales have teamed up to present to you Celebrating Strong Women in Science as part of both the 7 Decades for 7 Days and the Garvan Public Seminar Series. 2021 is the 70th anniversary of the Science Teachers Association of New South Wales, and it is wonderful to be part of this week, coinciding with National Science Week, to be part of the celebrations of scientific and education advancements, particularly those here in Australia. We have a long history of remarkable science and scientific impact. I'm particularly thrilled to be joined by Dr. Lauren McKnight. Lauren is both the Vice President of the Science Teachers Association and one of our science educators here at the Garvan Institute within the Kinghorn Centre for Clinical Genomics. Lauren, I'd love to welcome you just to give us the official welcome, please. Thank you.
Thanks, Mara-Jean. I'm very excited to be part of both the organisations that have partnered together today. Welcome, everybody. It's so lovely to see you all here. So the last 70 years has been an incredible journey for the Science Teachers Association of New South Wales, and it's also a very long time in science. There have been some incredible progress made during the last 70 years in genomics, the DNA research that we are so strong at Garvan, in immunology and in cancer research. And behind each scientist, here at the Garvan and also around the world, every one of them will have a story of the science teachers that inspired them, that taught them, that gave them that fundamental understanding of how the world works, that they have then driven forwards in their careers. We believe in supporting science teachers. We believe in their importance. We believe in equipping them and inspiring them to be that inspiration for the students of the future who are going to step into that role of progressing our knowledge. So this is day 3 of our 7 Decades in 7 Days 70th anniversary celebrations. We are holding it in the virtual space. We have an incredible platform that we are using to bring everybody together, to bring teachers out of their labs or their virtual teaching spaces at the moment, to bring them and the publics and the families from around New South Wales to come together to celebrate everything that science is in Science Week. We join with the arts communities, the technologies and innovations, and we join with all the very many diverse communities that make up science and that make up Australia. And we can celebrate this great strength that we have in science in Australia and in science education. And today in particular, we are celebrating the women that are part of those communities. Really looking forward to hearing from each of our wonderful strong women speakers today. Each of these women that you will hear from are doing incredible pioneering work in medical research. For the teachers that are here joining us from our 7 Decades in 7 Days event, we do have a Garvan stand in our virtual exhibition hall, and there you can find some of the resources that we've produced here at Garvan about genomics and some about our other research that we do here. So I will join these wonderful scientists at the question and answer that we have at the end of the presentation. Thanks, Mara-Jean.
Thank you so much, Lauren. Now, before you disappear, I'm going to put you on the spot and ask you a quick question. This is such a challenging time for everyone, and particularly for teachers and parents homeschooling. What are you hearing from your science teachers about how they're navigating this time in our history?
Look, I think teachers are some of the unsung heroes of the pandemic. The way that they have been able to pivot multiple times to multiple different modes of learning has been truly incredible to watch. I'm particularly inspired by the way that teachers have come together online and just sharing so generously with the resources that they have. It really has been a incredible cohesive movement, and they all deserve a medal and definitely a pay rise at the end of this.
I couldn't agree more, Lauren. Thank you for that. And a huge shout out to all our teachers and especially our science teachers. You guys are doing a phenomenal job. So thank you so much, Lauren. Now, before we hear from our remarkable speakers today, I'd like to talk you through some quick housekeeping. We will have a live Q&A at the conclusion of our talks. For those of you unfamiliar with the Zoom function, you will find the Q&A double speech bubble at the bottom right hand side of your screen. We would encourage you to enter your questions as they arise throughout the session, and I will do my very best to get to those questions throughout the course of the live Q&A. If you have any specific questions that we don't get to today, or that you perhaps don't want to ask quite so publicly, please do post them into the survey at the conclusion of today's event, along with your name and email address, and we will do our very best to come back to you one-on-one with a response to your questions. So, to our first incredible speaker, Sarah. I'm delighted to introduce Sarah Alexandrou. Sarah is a postdoctoral research officer who has just finished her PhD, congratulations, Sarah, in the Replication and Genome Stability Laboratory at Garvan. Sarah is studying how cellular abnormalities could contribute to the development of therapeutic resistance in breast cancer, which impacts so many of us and our loved ones. Sarah, congratulations on getting your PhD in. That must, that's such a huge effort. How do you feel?
I was so relieved when it was done. It was such an exciting, also anticlimactic, because it was just when lockdown was happening, but I'm just so excited to have it done.
Well, congratulations again. The next time we welcome you into the Zoom room, we will be introducing you as Dr. Sarah. Now, please, over to you for your presentation. Thank you so much.
Okay. Hi, everyone. It's such a pleasure to be here today to tell you a little bit about how I became interested in science, how I became involved in medical research, and share some of the work I'm doing here at the Garvan. So I first became interested in science and the medical field after religiously watching shows like Silent Witness and NCIS when I was a teenager. To me, these shows had such a strong female presence in a lab setting that I was determined to have a career as a forensic pathologist or a forensic scientist. So with that in mind, when I was in high school, I did biology as a HSE subject because I knew I wanted to do a science-based degree at uni. And when I went to uni, I did a Bachelor of Medical Science. But during my first semester, I really struggled with the compulsory chemistry courses that we had to do. And I struggled so much that I was in the process of transferring to a non-science degree because I couldn't bear the thought of doing any more compulsory chemistry subjects. But I stuck with it and I scraped through chemistry, and I'm really grateful I did because I loved the rest of my degree and I got to learn about the human body and how it functions and how it responds to diseases. And a group of diseases that I became interested in was cancer. And this led me to do an honours project at the Garvan in a group that was headed by Dr. Liz Calden. During my honours, I got to work with 3 brilliant scientists, Christine, and Liz, who were so passionate and dedicated and who I learned so much from that the following year, I started my PhD in the same lab. During my 4-year PhD, our lab expanded and I now get to work with such an empowering group of scientists at different stages of their science career. And as Mara Jean just said, I submitted my PhD in May and it was excitingly awarded last month in July. And the focus of my PhD was on breast cancer research. Now, breast cells, like all the cells in our body, go through a life cycle. And the difference between healthy cells and cancer cells is the rate of cell division, how quickly and accurately they go through this life cycle. Happy, healthy cells divide with absolute precision. And if there's problems along the way, the problems get fixed. And if the damage doesn't get fixed, the cells then enter a program of cell death. Cancer cells, on the other hand, have an uncontrolled cell growth pattern. The cancer cells don't respond to anything along this cycle telling them to die, and so they keep growing. When we relate this back to breast cancer, it's the cells within the breast that are dividing out of control. And why is this important? Well, breast cancer is the most frequently diagnosed cancer in Australian women. In their lifetime, 14 out of 100 women will be diagnosed with breast cancer. And while the 5-year survival rate of breast cancer has significantly increased in the last 40 years, we still have a situation where 3,000 people will die each year from breast cancer in Australia. One of the subtypes of breast cancer, and it's the subtype I've been working with, is driven by hormone receptors. This hormone receptor positive breast cancer can be treated with 5 to 10 years of targeted hormone therapy. And while hormone therapy improves the survival rate of patients, one of the biggest problems we face is cancer recurrence. So this recurrence is a major issue in breast cancer. This happens when the breast cancer cells find ways to bypass treatment and grow despite the therapy being there. These cells can then spread from the breast to different parts of the body, like the brain, the liver, and the bones, making treatment much more complex and contributing to those deaths. One of the drugs I got to work with during my PhD was called palbociclib, and this is a new type of drug that has recently been approved for use in breast cancer. This group of drugs prevents the cancer cells from completing their life cycle. In patients, palbociclib, in combination with hormone therapy, is very effective at controlling the progression of recurrent breast cancers by 1 to 2 years. But after that, the cancer cells stop responding to the treatment. So this is where my PhD project came in. I was looking at breast cancer cells that no longer responded to hormone therapy and palbociclib, and I was trying to create models to better understand this disease and come up with new ways to treat recurrent breast cancer. One of the ways I identified how breast cancer cells were becoming drug resistant was by using a gene editing tool that cuts out every gene from the genome, one gene at a time. We can then treat these cells with hormone therapy and palbociclib and sequence the DNA from these cells with the genes cut out to identify which genes drive drug resistance and cause those recurrent breast cancers. When we get the data back, we get long lists of genes that are involved in promoting drug resistance. And what we can do is we can put those lists of thousands of genes into specialized programs that allow us to visualize which genes are linked together. And what we were able to find is that breast cancer cells that are drug resistant have lost a core group of cells that are involved in initiating cell death. One of our challenges was then trying to identify what the best ways of treating these breast cancer cells that had lost these core cell death genes. One way we're doing it is by using drugs that are used to treat blood-based cancers like leukemia. And these drugs cause cell death in cancer cells. And what we've been able to do is we've taken these resistant breast cancer cells and treated them with a vehicle control, a hormone therapy, palbociclib, and our leukemia drug by themselves. But we find that the cancer cells are still able to grow. But when we combine all three drugs together at different concentrations, we find that the resistant cells start dying. And this triplet combination is really important because we're targeting the multiple pathways that the resistant breast cancer cells are using to bypass therapy and survive. So to pull everything together, about 1 in 3 patients treated with hormone therapy will stop responding to treatment, and that cancer will progress to a recurrent breast cancer. But what we found is that these resistant cells have lost these core cell death genes that allow the cancer cells to survive and adapt to therapy. One of the most important things we do in the lab is to take our findings and translate them into a clinical setting where it has the potential to impact patient outcome. To do this, we're collaborating with clinicians and scientists who have access to samples which have kindly been donated by clinical trials participants, treated with hormone therapies and palbociclib. And we're screening those samples to see if we can identify a subset of patients who have lost these cell death genes. We're also testing our triplet combination therapy in different models of breast cancer with the goal of designing a clinical trial to test this triplet combination in this subset of patients who have lost the cell death genes, with the hope of improving Australian breast cancer care for patients with recurrent breast cancer. So with that, I'd just like to say a massive thank you to everybody involved in this project, especially my supervisor and Christine, and say a really big thank you to everyone who donates samples from clinical trials participants and all the donors and the funders who make this research possible. Thank you.
Sarah, thank you so much. That was such a wonderful presentation. And I think the audience will agree we are in safe hands for our future scientific leadership with you in the mix. Really amazing. I do have a quick question for you, which is somewhat selfish. Was the photo in that last slide, was that the Paddo Bowling Club, lawn bowls?
That was our Christmas party last year. I think we went to North Sydney.
It looked amazing.
Sorry, you go.
May we lawn bowls again.
I know, I think it was the last time our lab was, all together.
Something to look forward to once the vaccination rates are up. Sarah, thank you so very much once again. I'm thrilled to now introduce our next speaker, Associate Professor Elissa Deenick. Elissa is head of the Lymphocyte Signalling and Activation Laboratory here at the Garvan Institute. Her research is focused on lymphocyte activation and differentiation and how this is controlled to ensure protection against infection while avoiding immune responses like allergy and autoimmunity. Elissa, welcome. Thanks for joining us.
Thank you. Thank you so much for having me. I love to be able to talk about immunology and how our immune system protects us from disease. But before I do that, I'm just going to run you through a little bit of how I got to where I am, because people don't always understand what the path is to becoming a medical researcher. So I started off doing a Bachelor of Medical Science, somewhat like Sarah, and like her, I did honours, which meant that I spent my 4th year in the lab doing immunology research. And like her, I loved it so much that I decided to commit to another 4 years doing research for my PhD. Again, over at Sydney Uni. Now, when I finished my PhD, I did what many medical researchers do, and that's to go overseas. And we go overseas because it means we can get exposed to new ideas and new techniques and kind of advance our science even more. So I went to Canada, to Toronto, to work with an amazing scientist, Pamela Ohashi, who had done a lot of work on autoimmunity and who I really wanted to work with. And after 4 years working with her, I returned to Sydney and to the Garvan Institute, where I've been ever since. And about 5 years ago, I started running my own research group. But I've left out an important part of the story, which is what inspired me to become a medical researcher in the first place. Well, I've always liked science, and my dad was a science teacher, so that might have some role in it. But I really think the person I really have to thank is Mrs. O'Sullivan, who actually I think is online today. So thanks, Mrs. O, if you are listening. And she was, when I was in high school, she organised the extension program. And she organised for me to go to a science summer school when I was in year 10, I think. And at that science summer school, there was a whole lot of stuff about human biology. And I just realised how interesting the human body was and how complex. And I really wanted to learn more about it. And so that set me on the path that at the end of year 12, I decided to do medical science. And then as I did that degree, I fell in love with immunology, because if you're talking interesting and complex, the immune system really meets those. So, and part of that reason is because the immune system has a complex job. So the role of the immune system is to protect us from infection, but there's lots of different things that can infect us: virus, bacteria, parasites, fungi, and the immune system has to respond appropriately to each of those to clear the infection. But what is our immune system? Well, basically it's our white blood cells. So if we were to take our blood and separate it into its different components, about 1% of it would be white blood cells or immune cells. And there's not just one type of immune cells, there's many different types of immune cells that work together to effectively clear the different infections that we encounter. Now, usually that works pretty well, but some people, their immune system is damaged in some way and they can't fight off infection properly. And we call this immunodeficiency. And there are many reasons that you might end up with immunodeficiency. The very old and the very young tend to have poorly functioning immune systems, things like HIV or immunosuppressive drugs. But what I want to talk to you about today and what I work on is genetic causes of immunodeficiency. So, of course, each of our cells has DNA, and that DNA carries the instructions for cells on how to function. But we can get variants in that DNA which kind of mess those instructions up. And if the part of the DNA that's got the variant in it has instructions that are important for immune cells, that can affect our immune system and its ability to fight infection. Now, if you're like me, over 40, probably the best known person with a genetic cause of immunodeficiency is this boy, David Vedder, or the boy in the bubble. So he had a variant in his DNA that basically meant that he had no immune cells, which meant that he couldn't fight off any kind of infection. And so from the moment he was born, he was placed in a plastic bubble to kind of protect him from all the infectious things in the environment. Now, most immunodeficiency isn't that severe. People still have some immune cells, but they may just not function quite properly. So what we've got to understand is that when we encounter an infection, say a virus, the outcome of the battle between our immune system and that virus is going to be determined by a number of things. How fast our immune system responds, kind of how many troops it can rally to the cause, what kind of weapons it has to fight that virus, but also what kind of weapons the virus has to try and fight back and protect itself. So really we've got to, and any defect in the immune system can kind of shift the balance from clearing the infection and getting better to an infection that's really serious. So I want to tell you about one group of patients that I've been studying, and they have a condition with a long complex name called activated PI3 kinase delta syndrome, or APDS. And these patients have a problem with particular infections. So they get frequent bacterial respiratory infections and pneumonia, frequent bacterial ear infections, as well as severe or persistent infections with just some viruses, not all, but some. And we know that these patients have a variant in their DNA, which is the instructions to make a protein that's really important in kind of immune cells sensing their environment. But how does this kind of change in that sensor mean that these immune cells now can't fight off these bacterial infections? Well, I don't have time to tell you about all the work we've done, but I'll just tell you about work we've done on one immune cell, and that's the B cell. So B cells are one of our immune cells, and their role is to produce antibodies, which are these little Y-shaped molecules that you can see here. And basically antibodies are pumped out from the B cell and they circulate in your blood. And then if they find the bacteria or a virus that they bind to, they can stick to that and help eliminate that infection. So how do we look at the B cells in these patients? Well, basically we can take their blood, separate out the B cells, and then in the lab, we can kind of culture those B cells and see how they respond to different signals and if they respond normally. And we also have these kind of sophisticated machine. This is a flow cytometer, and that's Julia, who works in my lab, where we can kind of look at the features of the cell, like at millions of cells at a time, and say, do these B cells respond normally? But of course, putting an immune cell in a little pink liquid in a dish isn't quite the way it usually operates kind of in our bodies. So we also developed a mouse model where we were able to put the same variant in the DNA that we see in the patients into the mice. And then we can immunize those mice and study how their B cells respond and really understand what goes wrong. And so we did this for several years. And what we discovered was that, unlike the boy in the bubble, these patients do have B cells and they can produce a bit of antibody, but they produce kind of lower quality antibody than normal people. So why does that matter? Well, usually if we have bacterial infections, part of the way that we clear those infections from our body is through another immune cell called macrophages, which basically means big eater. So these macrophages can kind of gobble up the bacteria and break them down and therefore clear them from our body. But remember I talked about earlier that some bacterial viruses have ways of fighting back against our kind of immune system. And there are some bacteria that have kind of this coating on them, what we call a capsule. And that kind of makes them slippery in a sense, so that the macrophages are unable to eat them up. So we can't clear them from our body. But that's where antibodies can come in. So if you have antibodies that can bind to the bacteria, then the macrophage is actually able to grab onto the end of that antibody and therefore gobble the bacteria up that way. So in our patients, they can't make these good quality antibodies that can bind to these encapsulated bacteria and swallow them up. And that's why these patients get these recurrent infections, particularly with these bacteria, which have this coating on them. So that's great. That's really interesting. But what do we do with this data? Where to from here? Well, one of the great things is that now we have tests in the lab where we can check the function of these cells, and we know what the problem is, and we have a mouse model the same, we can now kind of start to test new drugs that might be used in therapies for these patients. Another important thing that we're seeking to do is that clearly we can't treat the patients if we don't know that they've got this variant in their DNA or some other variant that's also affecting their immune system. So we're also got this group Circa with doctors and scientists who are looking for these variants in the DNA in patients who get all these infections to try and diagnose them. And finally, actually, it tells us really important things about how B cells function normally and how we get B cells to make the best, highest quality immune response and antibodies, which is really important. If you've been following a lot of the information about COVID vaccines, we need high levels of good quality antibodies. And as we understand B cells more, we can make better vaccines. So thanks. Oh, and just a call out to the science teachers. If you're not familiar with this website, BioRender, that's what I use to make these little pictures here. And it's excellent and you can use it for free. So I highly recommend it.
Love it, Elissa. Thank you so very much. And I'm going to pop you on the spot very quickly, if I may. So for those in the audience, Elissa runs her own laboratory, as she's described, and she's shared that incredible research with us today. She's also the chair of Garvin's faculty. And so, Elissa, given that today's session is all about strong women in science, and given that we've hopefully got teachers and future scientists in the audience, both in the Zoom room and on live on Facebook, what would you say to the younger generation thinking about starting a career in science, particularly the young women?
Oh, I love it. I think, what other job can you say that you get to find out new things about the body that no one has ever known before, and at the same time you can help people hopefully lead healthier lives? Like, it's just the most fascinating place to work in, and if you're interested, just go for it.
Amazing. Thank you so much. And a shout out to your dad for helping inspire you.
Thanks.
Thanks, Elissa. Now it's my great honour to introduce Professor Susan Clark. Sue is, I hope she'll forgive me for saying so, a national scientific treasure, a globally recognised pioneer of epigenetics, the study of all of the information encoded within and on top of the DNA that organises and regulates gene activity. Professor Clark's research has helped revolutionise the field through new technologies that profile DNA changes both in early development and in one of the diseases having the biggest impact on our society, cancer, particularly breast cancer and prostate cancer. And Sue's work has laid the groundwork for using epigenetics to develop and monitor cancer progression. She's been recognised with many accolades. Sue, thank you so much for joining us.
Thank you, Mary-Jane. I'll just share my screen and we'll start from here. Great. You can all see that?
Perfect. Thank you.
Great. Look, thanks, Mary-Jane, for the very kind invitation and for the honour of being able to talk to you today. So I would like to just share with you a little bit about my career journey, but in particular, how it has mirrored our growing understanding of DNA and how we're trying to now harness that information to impact human health. So my journey is obviously a lot longer than our previous speakers, and I realized that I've been working in this area for a long time. I was born in Melbourne and then moved to Canberra in grade 4, and then proceeded to do my education at primary and high school in Canberra, and then went on to do a science degree at ANU before I moved to Adelaide University, and then went to Sydney to work as a postdoc in the biotechnology industry, and then moved to Cambridge to learn more about molecular biology before I returned to Australia, where I moved to Sydney and joined CSIRO as a research fellow to continue my work in molecular biology before I joined the Garvan Institute back in 2004. And that was really led by the award of the NHMRC Fellowship, which allows scientists to be independent and have a career structure. And I've been blessed to be funded by NHMRC and continue to be able to continue my research, which is now very much focused on genomics and epigenetics. I'm just trying to advance this, yes. So when I reflect on my career and I realize how my career has really been impacted by the advances in genomic technology. So if we look here, I was born pretty much just after the double helix had been described. I moved to Canberra in grade 4 when the genetic code was first described, and that was incredibly exciting to not just take the structure of the DNA, but to see that the 3 base pair could code for amino acids. I was in year 12 when gene cloning was first described, and I finished my honours degree when DNA sequencing had just been developed. And that inspired me to go to Adelaide to do my PhD in DNA sequencing, where Maximum Gilbert sequencing had just been developed in Cambridge, and Adelaide was the first place in Australia to implement that. And I was lucky to be one of the first PhD students to use DNA sequencing to look at human genes. Recombinant DNA technology, which was using gene cloning, and in particular how our first vaccines were being made, was developed in 1982. And that was what inspired me to then join the biotechnology company industry for 5 years, where I was so excitingly involved in recombinant DNA technology to produce vaccines. And that was a very exciting time of my life. I then decided that I needed to understand a little bit more about molecular biology. And interestingly, PCR was developed in the middle of my postdoc period. And you might know PCR, polymerase chain reaction, which is fundamental to many of our molecular biology advances. But in particular, it is the basis of the COVID-19 detection assay that we all know about that is being used now. I then proceeded to work and develop my areas in molecular biology, and in particular, understanding gene regulation. And this was inspired by the completion of the Human Genome Project. And my interest in that completion was that that didn't tell us everything about gene regulation. We needed to know more about it. And epigenetics is something that really has inspired me. And we developed methylation sequencing that I have continued my life's journey on being able to see how that impacts on gene regulation. Since I've moved to the Garvan, there's been the development of many of these genomic technologies that allows us to go beyond the understanding of the DNA, but to understand how that reflects gene transcription, chromatin changes, and 3D structure. Now, when I think about what are the role models that really influenced my career, and I've had many, but when I think about it, there was one key event that was really important in the choice of my career. And this is when I was in year 12 at Deakin High School. And I share you this photograph because it shows you really what an unruly lot of students the poor teachers had to deal with. This is me. I happened to be a school captain. These were my class at the time. And it was really, we were very free-spirited. And the teachers did have a challenging time. We're a very new school, and it was the first year, year 12 was there because Deakin High was a very new school. And one of our relief science teachers who came to teach biology for 4 weeks only, Mrs. Kirk, she was tasked with teaching us about photosynthesis, which was obviously a very interesting area. But she took it one step further, and she reached out to her colleagues at CSIRO in plant industry, where they were researching about chloroplast structure and function. Now, back in 1972, this was incredibly novel work. It hadn't even yet been published yet. And she enabled our class to go to CSIRO, and there the scientists talked to us about their experimental work, very new data about understanding the biochemistry of the chloroplasts and how that was informing photosynthesis. The scientists then got so inspired by our unruly group, they actually gave us experimental tasks to do. And for 2 weeks, we were involved in their research. And that was so inspiring, and I think totally changed my idea of what I wanted to do. And that then made me decide I wanted to do science. I actually enrolled in a botany degree and biochemistry so I could further this research on what happens inside a cell at the molecular level. So thank you, Mrs. Kirk, that you're an amazing influence of my life. So the key questions now that our lab has been working on really probably for the last 20 years is to further understand the interplay between our DNA sequence, which is called our genome, and this epigenome. And the epigenome we now know is the information that is above our DNA sequence. We know that our DNA is decorated by a myriad of chemical modifications, some of which are called DNA methylation, which is a carbon-hydrogen group attached to cytosine in the DNA, but others are proteins which can be chemically modified and wrap around our DNA. And it's the association of the epigenome to our DNA that really plays a major role in controlling gene expression. But the other insight that we know can control gene expression is the fact that our DNA isn't linear. It's not just a sequence of A, T, G, C in a linear space. But our DNA is organized in an exquisite way in three-dimensional space so that it fits inside the nucleus in an organized way such that genes can be expressed in different cells at different times. So another key question is we need to really understand the mapping of how this occurs in different cell types. And importantly, how does this go wrong in disease? And in particular, my main area of research is how this goes wrong in cancer. And then how can we use this incredibly beautiful but complex information to better treat patients? So if I zoom inside a cell and inside this three-dimensional space, what we now know due to these new technologies of being able to look at chromatin contacts is that for a gene, as shown here in a cartoon, to be activated, then regulatory regions called enhancers have to come into contact with the promoter of that gene in three-dimensional space. And enhancers can be kilobases away from the actual promoter, but they do need to contact to be able to activate that gene. And I show here the DNA is decorated by all these epigenetic modifications, which determine whether they're active or inactive. And one of the key inactivation chemical modifications is this DNA methylation, which I'll go on to talk about as demonstrated here by the red lollipops. So it's really an important discovery that our genome is regulated in such a complicated way. So my group, in understanding epigenetic control and how it influences the genetics, is we've been working on 3 different pillars of my lab's work. We continue to try and understand the mechanism, so that will lead us to preventing cancer. We've been developing biomarkers that allows us to look at ways of early diagnosis of cancer, as well as monitoring disease during treatments. And more recently, we've been seeing whether some of our epigenetic discoveries lend themselves to new therapies. And so this is what the vignette that I'd just like to briefly talk to you about today is our new work about the promise of epigenetic therapies to potentially treat cancer resistance. So the cancer that I'm going to talk to you today about is breast cancer, and it is breast cancers that are ER positive. Now, roughly 70% of women with breast cancer will be ER positive, and the rest will be ER negative, ER being estrogen receptor positive or estrogen receptor negative. Now, ER positive breast cancer actually has a really good prognosis relative to ER negative breast cancer, such as triple negative breast cancer. And that's because these patients can be treated with what's called endocrine therapy. And an example of endocrine therapy is tamoxifen treatment. And that's really well tolerated for women, and they can go on long-term therapy, and that basically represses the growth of the tumor. However, after long-term therapy, there is a proportion of women that will relapse. So as shown here, about 30%, unfortunately, of patients with ER positive breast cancer who have been on long-term, for example, tamoxifen treatment, will actually relapse around about 5 years or longer. And once they relapse, then treatment becomes more of a challenge. So our preliminary data, we've been looking at this to see whether we can understand the molecular mechanism behind behind what causes resistance to breast cancer. So as I said, long-term treatment of tamoxifen is well tolerated, whereas when they become resistant, they do not respond to tamoxifen anymore. And our preliminary data has shown that the samples that respond very well have these enhancer regions in these critical estrogen receptor binding sites are unmethylated, as shown by these white lollipops. And they form these three-dimensional contacts with very important genes to allow them to be expressed. But what we find in the resistant cells is that these enhancer regions gain DNA methylation, as shown by the red lollipops. And this gain of methylation means that the estrogen receptor, which is critical for gene expression, can no longer bind. And that results in a disruption of these three-dimensional enhancer promoter interactions and results in aberrant gene activity. So this was a very exciting finding. We've now gone on to hypothesize whether, with this information, whether we can use this new, it's called epigenetic therapy, which can result in loss of DNA methylation, to see whether if we can use this to reverse endocrine resistance such that these patients can go back onto endocrine therapy for a longer term. So our recent work, as we just heard from Sarah, is using these avatar type models where we take breast cancers from women and put them into mice. And this particular example, I'm just showing you metastatic samples from lung and liver that are resistant to endocrine therapy. And here we've treated these mice either with no treatment, we've called vehicle, or with this new endocrine therapy called decitabine. Now, decitabine is an FDA-approved therapy. It's currently being used for blood cancers such as leukemia, but is not yet used for solid cancers. And we're doing this in collaboration with the Stand Up to Cancer group in the United States, Epigenetic Dream Team. And what we've found is that indeed, this is the vehicle, no treatment, the cancers grow rapidly in the mice. But when we treat with the endocrine therapy, so the epigenetic therapy, we find that the cancer cell growth is substantially reduced, indicating that this therapy does have an impact on slowing the cancer growth. To work out what the molecular mechanism might be, we've performed many of our genomic technologies and epigenetic technologies to look at the effect of methylation, transcription, chromatin changes, as well as three-dimensional changes. And what we find is indeed this therapy results in a reduction of DNA methylation across the genome in the treated tumours. But very importantly, we find that this loss of methylation primarily occurs at what's called these enhancer regions. And so they're the targets of where DNA methylation is lost. And when we look at what influence that has then on the three-dimensional structure, what I'm showing you here is an example of a very important gene in endocrine resistance. In the vehicle treatment samples, this shows you the 3D interactions, and there's no interaction between the promoter and enhancer, so it's a linear structure. Whereas when we treat with the hypomethylation agent, we now see this direct three-dimensional interaction between the promoter and the enhancer region. And this corresponds to activation of these genes. So this data that we've been producing is supporting our hypothesis that loss of methylation can result in changing three-dimensional structures in these very important ER-regulated genes. And when we looked at metadata from many, many human studies, we show, for example, in this gene, that there's very much poorer survival when this gene is indeed methylated and not expressed. Whereas when we get this demethylation and expression, it increases survival dramatically. So in conclusion, I've just given you a vignette of the future of now looking at epigenetic remodeling as a key mechanism, in particular in this particular cancer. We've shown that it can suppress tumor growth by causing loss of DNA methylation. It results in a rewiring of our three-dimensional interactions with this important transcription factor, the estrogen receptor. And selectively, it can change gene activation by altering 3D interactions. So now we're continuing this work because this preliminary preclinical data has shown that there is evidence supporting that we can reverse endocrine sensitivity. And this is work that we're continuing to do with the Stand Up to Cancer team to start clinical trials. So lastly, I would very much like to thank my wonderful team, of which I've had the work that I've just talked about today is from Joe, who's a senior postdoc in the lab, Claire Sturziker, who's a group leader in the lab, and Elissa, who will just be starting her PhD in this area next year. And of course, our funding bodies, we could not do this work without the generous support of the National Breast Cancer Foundation, Cancer Council, and of course, NH&MRC. So thank you, everyone.
Amazing, Sue. Thank you so much. And that is just a small insight into what's possible for the future in terms of treatments through epigenetic understanding of disease. So this really is the cutting edge of medical research right now. Sue, thank you so much. Now, Sue, before I open up to the panel, and I am conscious that we're running out of time, to the audience, you've had a absolutely remarkable career of success and no doubt of challenges. What would you say to yourself, if you could, looking back on the 1970s? I loved that photo. What would you say? What would your advice be?
Look, I'd have to say, I was the only student in year 12 to do a science degree. And everyone thought I was mad. Because we lived in Canberra, everybody became public servants. And I said, go Sue, that I did not become a public servant. And as I said, I was so inspired by this particular one teacher to show me that not just the concept of science, but the fact we could understand how things work before anybody else could. And I love that. I'm a very competitive person. So being able to be first at understanding something is really inspiring. And so I just say to everybody, go for it.
Well, congratulations and Keep going. You've got more work to do from the looks of things.
I know that.
Thank you so much. I'm just going to welcome our speakers onto the panel very, very briefly. We've got some fantastic live questions coming through. So the first one relates to Sarah, your work, your important work in breast cancer. How long will it take to get from these trials to use in patient therapy? So from the research and the trials into the clinic.
Yeah, that's a really good question. So just as an example, the drug that I talked about, palbociclib, it had decades of preclinical work, and it entered clinical trials in 2009, and it got approved for use in America in 2016. So that's 7 years that it took. But the good thing about our work is that we're using drugs that have already got approval for use in different cancers, so we know they're safe for use. So it's just about combining these drugs together to see what happens in patients.
Thank you, Sarah. And one for you, Sue, if I may. Julie asks, wonderful, Sue, is there a time frame for this testing to begin?
The testing, so there is methylation testing. Unfortunately, it is all in America. We have this major problem of bringing these things to Australia for many reasons, but we're hoping in the next few years the testing can be done in Australia for many of the cancers. As far as the therapy, again, those trials primarily have to start in America because they've got larger patient numbers, but we can do the phase 1 studies here, which is the safety trials.
Thank you so much, Sue. I'm conscious that we've got 60 seconds to go, and I know that a number of you are joining this Zoom webinar from work on your lunch breaks. Please accept our sincere gratitude for you taking the time to hear about this work and this important topic around celebrating strong women in science and indeed National Science Week. We will have a short survey that's going to pop up onto your screens shortly, and we would be so grateful if you could respond with your full and frank feedback, positive, negative, we want it all. Regrettably, we will be in the Zoom room for some time to come, so please do tell us how we can improve this experience. I know a number of our Garvan donors, corporate supporters, partners for the future and advocates are already supporting us in the Zoom room today, but if you've not supported Garvan and you'd like to, please do consider supporting our incredible research and of course that of our remarkable science teachers. We look forward to seeing you at our next virtual seminar, yet to be confirmed, but do keep an eye out on our website, and certainly if you've already registered for notification from Garvan, we will be sure to send you an email. I can't sign off without thanking the team behind the scenes, and we don't do this enough, I think. So to all of our speakers, thank you so very much. To Bronwyn Tyrrell, who is the convener of this session. Remarkable work, Bronwyn. To Amy and Tom, thank you for your incredible and essential logistical support. To all of you in the Zoom room, please stay well, stay safe. Get vaccinated if you aren't already. Just going to put that out there. And we hope to see you again very, very soon. Thank you very much.